Ladies and gentlemen, dear colleagues, students, members of the press. Whoops, nobody from the press here yet?
Beach.
It's a pleasure to welcome you to Honors Week in general and to the presentation of our gifted students this past week, and particularly at this moment in time, Cadet John Winalski. I almost said McKelvey. I have to keep them straight. I call one John M and John W. Okay, they're twins almost. But in any event, it's always a delight when one introduces a senior first classman who is about to leave, but then there's a sadness there. It was a sadness when I introduced John M. the other day and John W. now, because they'll be leaving soon. But the good news is that they usually pass the baton to others in the research lab, the Z-Team lab as we call it, the university team. And so we're looking forward to the legacy that they leave and that we pass on. John Winalski has been with us in the Z-Team lab for 3 summers. And it's hard to think of him not being there next year. But he insists that he's going to medical school. And he has the papers to show that he's been accepted to medical school. He's going to Marshall University in his home state of West Virginia. And so he's very much excited about that. He's also in the early process of applying for his, I guess it was a health professions scholarship, in hopes that he may coerce the Army into paying 4 years of medical school and a stipend, and then owe his country and his service, which he would prefer to do. So we wish him luck there. John has some exciting work to tell us about the heart and heart problems, particularly arrhythmias that he'll explain to you in a minute, what that means. And I'm excited that when we publish his work this summer, that it will tell a story that will excite both the general science and the clinical community with regards to possible treatments of this malady. But there's more to John than this being a scientific nerd, because our students are not nerds, they are well-rounded. And it shows with the rank on his collars right now. One example of his latest feat was to organize 500 of his colleagues in a spring FTX, going out into the community and serving others, and I think that speaks well to VMI training. The other part that just blows my mind, other than him being a great scientist and junior colleague, is that in less than a year, he goes to the National Collegiate Championship for boxing in California. Can you imagine? Never having boxed before, right? And just decide, hey, I'd like to join a club sport. And in less than a year, he was in the championship fight nationally that he lost by that much. So unfortunately, he's only ranked 2 in the nation with regards to his weight. But biology is not to be contended with. Don't fool with a biology major. Not even you, Wu-Ji.
Yes, sir.
Because we have also a boxing champion. In addition to John, we have Evan Thompson, who won the national championship in his category as well. So be careful when you confront a biology major. But John's work to me is personally rewarding and exciting, and we will have a publication soon to reward our relationship together. And John's going to talk to you about the role of estrogen and nitric oxide in prevention of cardiac arrhythmias in the embryonic zebrafish, Danio Rerio. John.
Good afternoon. Thank you, sir, for that very warm introduction. It's been a pleasure and a lot of fun working with you. I just want to say thank you. Major Pants, Colonel Rowe, Major Smythe, friends, Callum, fellow S5 rankers. Thanks for coming out. I know it's Friday, and so we'll try and get through this as quickly as possible, and hopefully you guys can learn something here. So before I kind of delve into what it is that I actually did with my time with Colonel Turner and the Z-Team, I'd like like to talk about the significance of why it is important. And it's worth mentioning that heart failure is a major cause of deaths worldwide, and over 50% of heart failures come from cardiac arrhythmias and sudden cardiac failure. The American Heart Association really defines cardiac arrhythmias as any change from a normal sequence of electrical impulses causing the heart to beat too fast, too slow, or erratically. And that's going to be really important further on when I kind of start talking about inducing arrhythmias in the zebrafish. And so as Colonel Turner said, my work was with nitric oxide and estrogen, and as you can see here, nitric oxide, abbreviated NO, is a gaseous messenger molecule that we believe is intimately linked in heart health and may be protective against arrhythmias. And it kind of works with estrogen in the fact that it's an upstream stimulator, or estrogen is an upstream stimulator of nitric oxide synthesis. And what that means is when one's produced, subsequently the other one will follow. The main difference being estrogen being synthesized by the enzyme aromatase. So in 2013, the American Heart Association released this study. And what the conclusion of that was that premenopausal women are protected from heart disease compared to men at the same age. However, when a woman goes through menopause, completes menopause, and their estrogen production declines by roughly 90%, they are then just as vulnerable to these cardiac anomalies as men at the same age. So that kind of poses the question here, are estrogen and nitric oxide linked in perpetuating health? And remember that if estrogen production decreases by 90%, then it's a safe assumption that nitric oxide production will also decrease by roughly 90%. So a brief introduction, delving more into nitric oxide. It's synthesized by the enzyme NOS, nitric oxide synthase. And it's present in the body as a free radical. And what that means is it has an odd number of electrons. Most molecules, atoms in the body like to have even numbers. Nitric oxide has an odd number. It's the byproduct of catalyzing L-arginine to L-citrulline, so taking one amino acid and shifting it to another one. And it's present in 3 different variations in the body, which we like to call isoforms. NOS, neuronal nitric oxide synthase, INOS, inducible nitric oxide synthase, and ENOS, endothelial nitric oxide synthase. And you have 2 main metabolical functional pathways through which nitric oxide works in the human body. You have a soluble guanylyl cyclase dependent pathway that focuses on some monophosphate right here. And you have the pathway that is completely independent of cyclic GMP and soluble guanylyl cyclase that works through nitrosylation. And that's the pathway that I'm actually focusing on, and I'll tell you what that means in a bit more seconds. Excuse me. But as you can see here, this chart right here is an example of what nitric oxide is and its function. As you can see, it's the production right here and some basic functions in the human body. It's anti-adhesive, anti-inflammatory, anti-migratory, and all these functions are very important for maintaining a homeostatic cardiovascular system. So here is really the nitrosylation pathway, and what you see here is you have nitric oxide and a protein that really hasn't been altered yet. And when you go through nitrosylation with your sulfur group kind of binding to your NO molecule, what's going to happen is this protein is then going to be post-transcriptionally modified. And what that's going to do is it's going to actually alter the function of the protein and kind of change its cellular effects. So this is the pathway that I believe cardiac arrhythmias actually present themselves in the cardiovascular system. I think my hypothesis says that if you dysregulate the nitrosylation pathway, what you're going to do is you're going to open ryanodine channels. And this deregulation comes from nitric oxide and estrogen deficiencies in the sarcoplasmic reticulum. And ultimately, this condition results in the arrhythmias becoming present. So logically, it speaks to the fact that if you could block or close these channels through some mannerism, specifically chemically for us, that sarcoplasmic reticulum calcium levels would return to a homeostatic range and the arrhythmic phenotype would no longer be present. Actually, skip a slide. And this hypothesis was molded based off of several outside research conclusions. And the fact that we based this off was that increased levels of cardiac arrhythmias are the result of calcium leaks in the sarcoplasmic reticulum. And that the leaks come from these ryanogene channels. And you can see that here. You have your basic ryanogene channel with the sarcoplasmic membrane right here and the flow of calcium. So as I said, the deregulation happens because of nitric oxide and estrogen deficiencies, which goes back to what I was saying when women go through menopause and their estrogen production goes down, subsequently their nitric oxide production goes down, and then you might see arrhythmias happen. And if you could close these channels chemically, then you would reverse the phenotype and the heart would continue to function normally. So the model with which we kind of developed this whole thing with was the zebrafish. It's been here at VMI for a really long time. It's great because we have the facilities to handle zebrafish compared to some other laboratory animals. But it's really important because zebrafish have a short developmental cycle. And within 48 hours you can take an egg and it will go all the way to a fully developed zebrafish. And the ease of care and size really explains why they're a good organism to use. But the main reason that I personally enjoyed them is because they're translucent and you can actually see through them under a microscope. You can see their fully developed cardiovascular system, which is they use a closed-loop cardiovascular system, which is similar to that of humans. And you can actually observe the heartbeat, the blood flow down the tail, and then return up through systemic circulation back to the heart. And another main point is the fact that you can rapidly diffuse reagents directly through their bodily tissue. So we would just place them in the solution and the reagents would diffuse through and then exhibit their effects. So everything with science, you have to have a place to start. And I was starting with Cadet John McNally from the class of 2014. I kind of came in at the tail end of his experiments. And he was using this model where you would begin treatment 2 to 4 days post-fertilization, and you would gather your data roughly 8 hours post-treatment. And what he was seeing in his results is that his experimental groups would yield 60 to 100% arrhythmias. Well, this is great, fine and dandy because he's maximizing, his population. He's inducing 100% arrhythmias sometimes. But the problem is, science needs to be consistent. It needs to be reproducible. You need to be able to do the same experiment over and over and over and get the same results every time. And we just weren't seeing that with Cadet McNally's model with arrhythmias. So the first thing that I sat down and did with Colonel Turner was a nitric oxide dose response, where we tested various micromolar concentrations of neuronal nitric oxide synthase inhibitor, N-NOCI, and we would gather our results 8 hours post-treatment. So we would take the zebrafish, we would place them in these various concentrations, 10, 30, and 50. We would wait 8 hours, and then we'd come back and we'd look at the fish under a microscope and record their heartbeats. And what this chart right here displays the fact that on the y-axis you have the percent arrhythmias in population, and on the x-axis you have your concentrations. And as you can see right here, 50% proved to be the ideal dosage. So every fish that was in the 50 micromolar of the N-NOCI solution exhibited arrhythmias past their treatment time. And our results were confirmed to be significant using a z-test with our p-value being less than 0.05 and the error bars representing a standard percent error. So this wasn't where we needed to make the change in our model. And because as you can see here, I'm going to walk you guys kind of through a timeline. So around 130 to 140 beats per minute is the average, the normal, the healthy heartbeat beats per minute of a zebrafish. And so in our solutions, you can see that gradually over time, 50 micromolar will drop down to about 65 beats per minute, while 30 micromolar will only drop down to about 100 beats per minute, and 10 micromolar would really only subtly change. And then what you can see here is once the fish were taken out of the solution after 8 hours and placed in their normal rearing solution, they would recover. And that's because zebrafish possess the ability to kind of flush their systems, if you will. So we would take them out of the toxins, put them back in the normal petri dishes, and they would recover. And so as I said, this kind of wasn't where we wanted to shift, make a modification to our model because it really wasn't broke, so there was no need to fix it. So then we looked at a treatment time shift, shifting from treatment from 2 to 4 days post-fertilization to 5 to 6 days. And this was based off of literature which suggested that around day 5 is when the vagus nerve would fully develop in the heart. And this correlates with the development of a more complete sensory, motor, and autonomic nervous system in the cardiovascular system, as well as a more developed, more complete cardiovascular system. So we're simply just saying that the fish are more developed and they're going to be more responsive to their treatments. And when we did this, what we were able to see was that, again, using 50 micromolar of NOC treatment, 5 to 6 days post-fertilization, was that, again, we were able to observe 100% arrhythmias, but it took a longer treatment period. The treatment period basically had to double, going from 8 hours to roughly 17 and a half. But what we observed was the fact that the results were more stable. They were more reproducible. So time and again, we could place zebrafish that were 5 to 6 days post-fertilization in 50 micromolars of NOC and observe the fact that this population would be entirely arrhythmic. And our standard for arrhythmias was any heart rate that was under 112 beats per minute. So now I'm going to jump into a brief introduction on aromatase. And if you remember, that's the enzyme that's responsible for synthesizing estrogen. And it's found in various tissues throughout the human body. And as I said earlier, estrogen deprivation is a serious but common disorder in women who have undergone menopause, with their estrogen production decreasing by an average of 90%. So then this just to remind you, is referring back to our link of estrogen and nitric oxide. Does this link right here protect women from these arrhythmias that I have just shown to be induced by nitric oxide deficiencies? So we went ahead and used the same model we did with our NOC experiments. 5 to 6 days post-fertilization, 50 micromolars of aromatase inhibitor, AI, and we left them in there for 8 hours at least as the minimum time. We usually cut them off around about 18 hours, so again, in line with our NOC model. And what we observed and what this chart displays right here is that these results mimic NOC inhibition, with the entire population displaying the arrhythmic phenotype. So as you can see here, our 50% AI experimental group is exactly identical to our NOC group. So when you deprive fish of aromatase, you see the same results as nitric oxide deprivation, with arrhythmias becoming present. So now that I can basically kill my fish, the question was, can we save them chemically? Instead of just flushing their systems out, can we actually block these abnormally open channels and reverse the phenotype? And Colonel Bell suggested to us that we use dantrolene. It's a muscle relaxant which chemically blocks ryanodine calcium channels. And it's currently used to prevent malignant hypothermia, which is a genetic condition that when patients are exposed to localized anesthesia, their body temperature will rapidly rise. So they're given a dantrolene sodium injection, and that will restore their body temperature to normal. And it's also used to alleviate muscle spasms in patients with cerebral palsy. So we had to kind of a hiccup in this whole experiment, but we used the new zebrafish model, fish 5 to 6 days post-fertilization, and 50 micromolar of AI or NOC solution with a minimum of 8-hour treatment time. And we saw that the population, as my data supports, was 100% arrhythmic with the accompanying paralysis. So they're on the verge of death. They're not swimming. And what we did is we added a 10 micromolar concentration of dantrolene following the presence of arrhythmias. And what we were able to see was that the fish would we would see them right themselves, and then they would start to swim around, and that would let us know that it's time to check the heart rate. And that then confirmed that dantrolene had actually restored the zebrafish hearts to a homeostatic range. And it also confirmed that there are no deleterious effects on a healthy zebrafish. So if a healthy zebrafish is left in dantrolene overnight, 24 hours, 48 hours, nothing is actually, no adverse effects are going to be observed. And what this chart here shows is at the normal heartbeat range of zebrafish. Following the treatment, you can visually see that it declines to around 80 to 75 beats per minute, and then it jumps back up following the dantrolene treatment to right about the normal range. And so initially, this was a very rapid response. It was really hard to record. I had to have Julie Lozier record one population while I recorded the other population. And we observed that NOC and AI both respond to dantrolene in the same manner, that right around 11 to 15 minutes, fish exposed to dantrolene will make a rapid recovery before recovering entirely. And when you compare that to our control solution, our control washout of just flushing their system with the normal fluid that they spend the rearing period in, and you see that it'll take about 30 minutes. So the conclusion here was that dantrolene fish regained motility and corrected heart function faster than the washout. But if you left the fish in the ERS and the dantrolene over time, both would recover completely. And that kind of still didn't leave us entirely satisfied because, as I mentioned earlier, zebrafish can flush their systems. So we weren't sure if we were actually curing the arrhythmias that we had induced. The mechanism of action was still unknown. Are we actually causing the channels to be closed by using dantrolene and then causing the extra calcium in the sarcoplasmic reticulum to dissipate, or are we just removing the fish from the toxins and having them flush their system, and that is what's restoring their function? So again, on the advice of Colonel Bell, we went a layer deeper. We did a calcium tagging experiment where we utilized two populations of fish. We had our ERS control, and then we had our AI and NOC deprived fish representing our experimental group. And once the fish, we followed the new zebrafish model, so 5 to 6 days post-fertilization, fish were placed in 50 micromolars of aromatase inhibitor, which deprives their estrogen, or the neuronal nitric oxide synthase inhibitor, which deprives